Very warm welcome to Bio Library, and we're going to discuss these 100 topics for DBT JRF. You cannot afford to miss these topics. Also, do not blindly trust on only these 100 topics. You should study them. Also, you should also figure out on your own that what are the other important things for you. Like, you cannot miss these topics, but also these topics, only these topics cannot assure your selection, right? So DBT is dynamic, DBT is difficult, DBT is competitive, and just lagging behind by some 2 to 5 or 10 marks may give you category second, which is not of much use from the point of view of fellowship, from the point of view of pursuing PhD, right? So supplement your preparation with these 100 topics. Without much further ado, let's get started. So the first topic is restriction digestion. Questions are recurrently coming from this topic. So you have to know what are the number of bands which come after the digestion of a linear DNA or after the digestion of a circular plasmid. Okay. So first one is this. Second is endo-reduplication. You have to figure out what endo-reduplication is. And also mind you that I'm not going to teach you anything in this slide. Okay. So you need to figure out on your own what is endo-reduplication. Third is fragile X syndrome. Just Google it. Fourth, opsonization. Immunology people, just I think you guys know it. It's very easy. Statins, which are, let me tell you, they are cholesterol inhibitors. Then also you need to figure out that which is the enzyme which is inhibited by statins. Then comes zDNA, whatever you call it, left-handed helix. When does it form? Like, what does it demand from the cell? As you know that in a real time, the different conformation of DNA can exist in a single cell, right? So when does it happen that a DNA takes the conformation of zDNA? Then comes catalase. Read about detoxification, read about antioxidants. Then you should read about this enzyme. Questions have come from this. Talking about catalase, how can we leave this thing called as glutathione? Okay. And then comes xenobiotics. All three of them are connected. One, catalase, second is glutathione, and third is xenobiotics. And then comes methanol poisoning. If you are, if you read some newspapers, then there is a term called Rangeen Sharab. So that is what methanol poisoning is. And you also know that it comes under the phenomena of competitive inhibition. So what do you need to administer when you are facing a situation when a person is methanol poisoned? Okay, so you decide on your own. I'm just giving you a hint that will it be formaldehyde or will it be methanol or will it be ethanol? Okay, so then comes prions diseases. Like they can ask you about Kuru disease. So prions disease, very easy. Then comes aldolase. There was a question that what is the class of the enzyme aldolase. So talking about this, figure out different enzymes. And then comes CPG islands, that what are the positions where your cytosine gets methylated and what is its repercussion if the the structure or the modification is present in heterochromatin or the modification is present in the euchromatin region. Then syndromes, different syndromes, you need to remember all of them. Klinefelter Turner syndrome, all of them, just go through them once. Okay, in your different syndromes. Okay, and what is that condition in which an infant would not survive, like if that person is having only y chromosome, X chromosome is lacking, then the person will have less chances, in fact, that infant would not survive. Okay, then comes frequency of alleles, talking about Hardy-Weinberg equation, Hardy-Weinberg allelic frequency calculation. So learn this. Then LOD score, and when you talk about mutation, you can study about this lot score analysis, and what is the frequent value, what is the range of a lot score value. Also read about these things. siRNA, talking about the broader term that is RNAi. So study about RNA interference. There was a question that in RNA interference, what do you use? Duplex DNA, duplex RNA, RNA DNA hybrid, or none of these. Okay, so you decide for yourself what it should be. Then comes SNP. SNPs are very important. See, it's an exam more or less concerned with biotechnology. So every term which comes under your molecular techniques as well as technology should become important, or is like naturally becomes important for the exam. So you have to study single nucleotide polymorphism. Then comes rabies. There was a question about Negri bodies which are formed in the brain of a person who is infected with this rabies virus. Okay, then comes Alzheimer's disease. From Alzheimer's disease, again the question was, what is the aggregation, what are those neurons which are lost, I think, in this. Those are cholinergic neurons, but you need to reassure yourself via googling it. Okay, so Alzheimer becomes again important. Then comes Tay-Sachs disease. Read about this disease, read about metabolic deficiency diseases like phenylketonuria and all that. See, whatever have come in the past question papers becomes important because DBT repeats its questions. Okay, then comes reporter essay like GFP analysis. What are the reporter tags which are used? What is the organism from which GFP is isolated? Everything. Then comes haptens, about your adjuvants, Freud's complete adjuvants, Freud's incomplete adjuvant. Question have come like 2 to 3 times. It has repeated, I think, 3 times from this topic. Then comes gene libraries, cDNA libraries. उसके बाद biosensor is there. Biosensor से there was a question regarding your this machine which tests your blood sugar level. So what is the biosensor present in it, like what is the enzyme which is immobilized in it. Okay, then monoclonal antibody, extremely important because questions are coming again and again from this topic. Then ploidy induction, like colchicine, using colchicine. So how to minimize the natural time development lag, and how to get fast some transgenics. So ploidy induction. Then comes exitoxins. Exitoxin is it causes shellfish poisoning, and I've seen that this question has repeated in 3 to 4 times in the DBT exam. Okay, so it's a crustacean poison. It's called as exitoxin. And then genetically modified organisms, I think you should know which is the first transgenic that was formed. A little bit of difference between GMOs and transgenics, but you don't have to go into that detail. Read about which was the first transgenic, or the transgenic which is getting used here. What is the who's the father of golden rice, and then what are the genes which are present or inserted in golden rice, and how many sources have been used in golden rice production. Read about these. And then dissolved oxygen, biochemical oxygen demand or biological oxygen demand. So they'll ask you that there is a culture, a dissolved oxygen is getting depleted, and that those bacteria are hypothesized to be, so they are hypothesized to be aerobic because they are utilizing your oxygen. Then DNA RNA structure, it might sound very cliche, very common, that who studies these, like you people already know it, but I assure you that there'll be question from DNA RNA structure, Sanger sequencing, or your what do we use, PITC sequencing, that is Edmond degradation, so degradation sequencing again. And then comes question from fed batch culture. Fed batch culture, if you're from engineering background, that is from biotech background, then it's fine for you. Otherwise you need to know what are these cultures, what is fed batch, what is continuous culture, what is discontinuous. Discontinuous I think doesn't exist, but what are these cultures. And then comes vitamins. You need to study about the cofactors. Okay, this is very important, this is very common and very important for your exam. Viral vaccines you need to study, and I think they might ask you some adjuvant of this covaxin also, so study that also, like keep in mind. And then comes nude and skid mice. So what are the enzymes which are deficient in these mice. And also it was asked, I think, about non-obese diabetic mice. Nod mice is also there, so you check on your own what is nod mice. It was also asked in some of the paper. And viral diseases also, like what are the causal agents, what is their strandedness as well as their genome is most commonly asked, like they are the DNA or they have RNA as their genome, or are they plus strand, minus strand, what are they. So viral diseases. Then enzymatic regulation, you need to study what are the reversible modifiers, irreversible modifiers, like aspirin inhibits your prostaglandins, but then again it's not an enzymatic regulation. For enzymatic regulation, let's go to PMSF, like PMSF irreversibly regulates. So go to these enzymatic regulations. And then pathogen derived resistance in case of plants, read about systemic acquired resistance for plants, PAMP, MAMP in case of plants. And then QTL mapping, if you can study, you just read about QTL mapping. Biofuel, like Jatropha or some of the plants which are it is being encouraged, so just read an article on biofuel. And then comes your phosphoenothricin. I think question have directly come from this, that what toxicity does it cause or something related to this, you just read it. And TI plasmid, which I think you all must know what is TI plasmid, what does it cause, and what does RI plasmid cause, and what is Virulence factor, what is the role played by virulence factor, who nicks the DNA, who protects the DNA. So all these things, what is the mode of integration of this DNA into the plant. And then comes culture, like you have to remember the name Guha and Maheshwari, which is essentially very important. And also you need to remember that haploid culture can be created by this. Then comes your artificial seeds. I also posted one question in video format in my Instagram account. So why do we use alginates in your artificial seeds? That is, somatic embryos are coated with alginates, right? Matrix of alginates. So yeah, artificial seeds. And then the next question is suspension culture. Say for example, you require a lot amount of vitamin B12. What do? Like, which experimental procedure will you follow out of the tissue cultures? So suspension culture will be the answer. So you can read this. And then not factors, like nitrogen fixation, when you'll read it, you'll remember this also. NIF genes, I think, yeah, NIF genes are also important. And then comes phototropins, cryptochromes, phytochromes. Remember all three of them. And what is the similarity, dissimilarity as to who absorbs red light, who absorbs blue light. So just this. And then secondary metabolites, extremely important, because it was the sulfur containing secondary metabolite, which was glucosinolates. And then the first alkaloid which was discovered, I guess that was morphine. So just read about these. Then comes nitrogen fixation as to whole, like NOD genes, NIF genes, nitrogen fixation. You have to study. They sometimes just ask you that what is NMOX, right? The questions are simpler, but again you need to read it so that you can be assured of the answer. And then comes alternate oxidase, like what happens then when there is stress to a plant, or what is this pathway. Cyanide resistant pathway, I think this is. And then comes your Calvin cycle. And there was a question that which the plant has both the cycles, C3 as well as C4. So it is very natural that a plant undergoing C4 cycle will have C3 cycle in it. Why? Because in C4 cycle, bundle sheath cells, inside the bundle sheath cells, your Calvin cycle is going on, right? So C4 includes anyway C3 cycle, right? Cam also includes anyway C3 cycle. So just remember that maize is a plant which is a C4 plant, and it has both the cycles going on, Calvin cycle as well as, C3 cycle as well as C4 cycle. Then comes your ABC model. Sometimes they ask what is the function of E gene, which is constant in all A, B, C genes, okay? And then comes your vernalization, that what are the genes involved in vernalization, what is we are in 1 2, and what is its function, what is its relation with FLC gene. And then comes plant hormone, just read superficially. They can ask you about receptors, they can ask you about DILA proteins, they can ask you about the functions, they can ask you about the ratios, whatnot, but not very deep. So go superficially through the plant hormones. Then digestion of protein, which I've already discussed with you, that this becomes important. All the sequencing thing becomes important because that comes under biotechnology, okay? And then comes alignments from bioinformatics. If not more, then you can read about global and local alignments. And DNA barcoding, this question comes as it is. Read about DNA barcoding. Pedigree, if you can solve it, very good. Otherwise you don't have much time, but I think you can solve it. The questions which come in DBT are way simpler than the questions which come in CSIR, okay? So pedigree, yes, you can give it a try. And then blotting techniques, like I think all of you must be knowing this snowdrop. This is a very like sweet trick, I would say. So S stands for southern blotting is for DNA, northern blotting is for RNA, and W is western blotting is for proteins, okay? So what will be the different blotting techniques? They'll ask you about some experiment if they're expressing mRNA. If they're talking about the quantification of expression, if QRT-PCR is not there, just make sure that you tick western blotting, do not tick northern blotting there, okay? So yeah. And then comes to, okay, what did I tell you? Did I tell you not to tick? Okay, so you need to tick northern blotting when they are asking about the expression only. When they are asking about you the expression only, then tick northern blotting. When they are asking about the quantification of the expression, again tick northern blotting if QRT-PCR is not there, okay? I'm so sorry I talked. So the idea is that you read about the mRNA in order to see the expression, how much of the genes are expressed. So you will see about the level of the mRNA. So you will do northern blotting, okay? And then comes ESTs, express sequence tags. What does it refer to? And it has come, I think, as it is. Then comes ELISpot assay, which is important. Cytokine, there are a number of cells, I think, which produce your cytokine. So T cells, which the frequency of T cells that produce cytokine, and this is quantified by ELISpot assay. So this is the question which has been repeated again and again, year and year. But you need to read it more about it, because they might ask you something else. And cDNA library. Then what is the advantage, what is the disadvantage, like what are the limitations of cDNA library, types of PCR and PCR. Just read about your pyrosequencing, that is emulsion PCR, okay, which is important. And then in PCR also read about your primers, that what concentration, what final concentration of primer will you use in a 25 microliter reaction if you are given with the conditions of 100 microliter reaction, okay. So the question will considered with mole. So types of PCR and PCR also read about how to calculate the primary concentration. Then comes DNA markers, dominant, codominant markers. Again, hypersensitivity. What are the different antibodies involved in hypersensitivity is more or less asked by the DBT time and again. And then comes antibodies. So different antibodies, which of these are soluble, which of these are membrane bound. Read about this, which comes in the first response, which comes in the later response. Then B cell, as it is, when is it called plasma cell, and what is required for its activation. And then comes elicitors also they are asked as it is. Then comes immunity in plants, elicitor, immunity in plant, that pathogen derived proteins, all of them are linked. So read immunity in plants, because questions are being asked. So enzymes of RDT, I think you must have studied it in the if I talk about the priority, then it should be your priority that you read about enzymes of RDT. Polymerases, what are the enzymes which are used in the laboratories, all these enzymes you should be aware with, okay. So then comes your codon, like they ask you if the code was made up of two nucleotides, and if the code was made up of three bases, four bases, three bases, I think we all of us know. What about the if the codon was made of four bases, what if the codon was made of two bases. So these are the different questions which are asked. I think degeneracy of codon has also been asked in the DBT. And then comes tRNA, extremely important, extremely important. So just read about tRNA, okay. What are the base modifications present there, and what is the angle between different arms of tRNA, and what is the orientation if some codon is present over your mRNA, what will be the anticodon present on tRNA, extremely important. And then come enhancers, extremely important, because they ask you about that, does it work in position dependent manner, is it cis acting, trans acting. So you need to read about this also. Heterochromatin and euchromatin, HC and EC, this I think all of you know, but read it with respect to the modification, epigenetic modifications. Then comes gene families, orthologs, paralogs. Like paralogs are formed because of duplication. Say for example, they are asking you about two genes, one in a human and other in a chimpanzee. Just read what have they given. Are they talking about analogy? Are they talking about homology? So just read about gene families. It becomes again an important topic. And then repetitive DNA, like microsatellites also they are asking. So read about it. And VNTR, which one the smallest repetitive DNA. Sorry, it is repetitive, therefore it I think it repeated, I'm sorry. And then oncogenes, and they ask you about, with oncogene comes tumor suppressor gene naturally, that what is haplosufficiency, and what is the mutation that is required in case of your oncogenes, from proto-oncogenes to oncogenes, and what required in your tumor suppressor gene to lose its function. Then comes diffusion. See, talking about oncogenes, proto-oncogenes, tumor suppressor genes, what are the mutations required, it looks like it's very easy, right? So you might get confused in the exam, therefore it's better to read it in this first-hand manner. And then comes diffusion. Along with diffusion comes your facilitated diffusion, the graph of facilitated diffusion. And then cell organelles, very important. I think they're asking about they're asking about cell organelles in a very applied manner. Say for example, if you have injected some dye into ER, what will be the different organelles which will be labeled. So the answer was your ER Golgi as well as nuclear envelope, because with the ER, nuclear envelope is also continuous. So cell organelles in an applied manner. Microscopy, very important, just read it throughout. And then centrifugation, yes, they ask you RPM and some of the formula which I discussed in my formula video, that in STS page. Say for example, one of the compartment is labeled, so what other organelles will be labeled. Read about your endomembrane system. And also microscopy entire, centrifugation entire. So centrifugation, I also mentioned the formula in my formula video. Just go through it, like how to convert RPM into, I think, radians per second. So that was the question. And then ion exchange chromatography, they ask about cation and anion exchangers. What are those? SDS page, every now and then they are asking about, say for example, you have run a gel, and they are comparing two of the techniques. One is your extraction chromatography, gel extraction, which tells you about the size of the protein. They'll perform your size exclusion, and here they'll perform your they will perform size exclusion, and then they will perform your SDS page. Then they'll ask you what is the nature of your protein. Okay, enzymology completely, please read what happens to Km and Vmax, and how to calculate Vmax, Km. Order of the reaction, unit of order of reaction, I again mentioned in my formula video. Free energy, again mentioned in that video, but if you don't have time to go through it, just read what is the relation between free energy, enthalpy and entropy. Then comes the type of DNA. Again, I'm talking that this sounds very cliche, but all these simple topics, they give weightage to the DBT exam. Okay, then comes Ramachandran plot. I think I've posted some question on Instagram. I will. Okay, so what does Ramachandran plot talks about more? As to, does it talk about what are the disallowed regions in your plot, entire plot? Okay, or does it talk more about the secondary structure? So Ramachandran plot. Then chemical bonds in carbohydrates, very important, because they can ask you what are the bonds involved in cellulose, like cellobiose is the monomer for it. And then they can ask you inulin. They can ask you something like not so convenient, but then also go through the trivial ones. So then amino acids, very important. Just read what are the polars, what are the non-polars, what are the amino acids carrying positive charge, negative charge. Just go through the side chains once. Bond energies, in the sense that you should know which are the bond will be strong. Also in the biological interactions like hydrogen bond, covalent bond, hydrophobic interaction, van der Waals interaction, which one will be having the highest energy and which one will be having the lowest energy. Try to learn how to convert joule into calories. And then comes pH calculation, Henderson-Hasselbalch equation. Again, if you type it in Google, you'll get to know that what is the equation and how to calculate it. Average, very important. Mean, how to calculate mean, again mentioned in the formula video. And series, I think also mentioned in the formula video, also mentioned in the last video, that series becomes an important part. Molarity stock solution problem, which anyways you'll have to do when you'll work in a laboratory. So yeah, it's important for the exam as well. Molarity calculation as well as the molarity stock solution problems, both of them become extremely important. Again, as I told you earlier also, because you all are putting in hard work, but self-belief is what makes the difference. Okay, so do not screen out yourself in these 2 days. You have just 2 days. Give your 100%, give all you have, and I think you'll get something. If not anything, then resilience. So with this, everybody, I take your leave and all the best for the exam. This may or may not be my last video before your exam, because I talked, or like I told you that I'll upload one more video regarding bioinformatics, but it's becoming hectic. But if I'll have time, I'll do it for you. Let's see how many years can I do. So I've done one thing, I have analyzed everything for you, but I have to make this PPT and all. So okay, let's see how it goes out. So thank you, and once again, just take the screenshot of this video, and I think 100% is gonna help you. One more thing, one more tip from my side, just read about Barney's bar star system. Okay, many times question have come from this, and it comes naturally, it comes. So read about Barney's bar star system. Thank you everybody, and keep studying.